Good afternoon, everyone. Welcome to the Quantitative Seminar. We'll get started at 12:30. We'll just wait another minute or two. Okay. All right. Good afternoon, everyone. Good afternoon. Welcome to the Quantitative Seminar. Today, we're delighted to have Megan Feddern as our speaker. First, I'd like to begin with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. So as I said, Megan Feddern is our speaker today. She is a PhD candidate here at SAFS, the School of Aquatic and Fishery Sciences, in the Holt-Grieve Ecosystem Ecology Lab, and a NIMPS Sea Grant Population Dynamics and Ecosystems Fellow. Her research interests include food web dynamics, specifically how species interactions are influenced by changes in the physical and biogeochemical environment. Megan enjoys harnessing the power of chemical tracers to examine complex ecological questions, and her current research uses compound-specific stable isotope analysis of amino acids to understand changes in primary production and foraging ecology of pinnipeds in the Northeast Pacific using museum skull specimens. Megan is originally from the White Mountains of New Hampshire and moved to Seattle for graduate school after spending a few field seasons moving around the country and abroad. Outside of research, Megan enjoys anything outdoors, particularly new activities that get her outside of her comfort zone, particularly backpacking, snowboarding, and biking. For today's talk, if you guys have questions during the talk, please enter those into the chat and we will send those to Megan. Megan has agreed to keep an eye on that and field questions during the talk. And then at the end of the talk, there'll be a chance for a more formal Q&A at that time. So without further ado, I'll turn things over to our speaker. Welcome, Megan.
Thanks, and thanks to the Essington Lab for all their organizing this quarter. Sorry, my screen just popped up. I gotta move it over. So today I'm going to be talking about reconstructing pinniped trophic position using archival museum specimens. When I first submitted this talk, I wasn't really sure what I was going to talk about, but I'm actually not really going to talk much about biogeochemical indices today. I'm really going to focus on trophic positions, specifically of harbor seals here in coastal Washington and the Salish Sea. As a PhD student, I work really closely with both Gordon Holt-Grieve, I'm in his lab, and Eric Ward as well. So just to give you a brief overview about what I will be talking about today, first I'm going to just start with a really general introduction about how stable isotopes can be applied for ecological applications, specifically nitrogen and carbon. Then I'm going to talk a little bit about challenges in ecological stable isotope applications, specifically how in dynamic environments, changes in biogeochemistry and physiology of the consumers that we're interested in can really impact things like our trophic position estimates when applying those types of data. And then finally, I'll talk about a case study that's a part of my dissertation, looking at harbor seal trophic position in Washington. I'll start with talking about how we parameterized our equations to calculate harbor seal trophic position, and then I'll talk a little bit more about the actual ecological applications of this work, specifically looking at how harbor seal trophic ecology responds to environmental change in prey availability over the last century. So just to make sure everyone's on the same page about some of the lingo and symbols that you're going to be seeing throughout this talk, I'm going to just introduce you to both nitrogen and carbon as chemical tracers in ecological environments. So first, nitrogen is used to calculate consumer trophic position. I'd say that's probably the most common application for nitrogen stable isotopes. And this symbol here is basically referred to as delta N, or a delta value. And what it refers to is basically the ratio of the heavier to the lighter nitrogen. And we standardize that over some standard, a universal standard for nitrogen. It happens to be air. And then we write it out in this per mil designation, which is essentially similar to a percent. So it's essentially a ratio of ratios, you can kind of think of it as. So what we can use then is we can take the delta N value from a consumer, we can subtract the delta N value of the primary producers of the food web that that consumer exists in, and then we can divide it by what we call a trophic enrichment factor. Now, the trophic enrichment factor is basically how nitrogen stable isotopes change as they move through the food web. So what ends up happening when a consumer assimilates some resource into its tissues is there tends to be a preferential assimilation of the heavier nitrogen compared to the lighter one. And as a result, as these resources are passed on through the food web, we get a higher delta N value and higher consumers. So in contrast, we also can use carbon isotopes to really inform us about the differences in ecosystems. They're primarily used to calculate things or look at things like movement and foraging location and changes in carbon sources as well. So you can see here we have a map of the Gulf of Alaska, and we have pretty dramatic gradients in isotope values that happen, and that's primarily due to the amount of productivity in an area. So where more carbon is being sequestered by primary producers, we're going to have a different carbon stable isotope value. And as a result, we can use those values to kind of track where animals are moving and foraging throughout their life. Similarly, some different types of carbon sources tend to have different stable isotope values. For example, carbon isotopes can be used to kind of differentiate between terrestrial or marine-derived carbon, or similarly C3 versus C4 plants. So we can use them to use how those different resources are getting into the food web. So now that hopefully we're all on the same page with some of this information about stable isotopes, I'm going to talk about some of the challenges with stable isotope applications, specifically how changes in biogeochemistry and physiology can really impact the values that we're measuring. So first and foremost, it's, as I mentioned in the previous slide, it's really important to measure this isotope value of primary producers when we're trying to calculate trophic position. The isotope value of primary producers can change based on things like nitrogen sources. So whether those producers are getting their nitrogen from nitrate versus ammonium versus urea, and also the isotope composition of each of those sources. Other things like light availability and taxa can also have impacts on those values. However, a lot of studies sometimes don't measure the stable isotope values of primary producers while they're measuring consumers, and instead they'll maybe use something from a different season or a previous study, or they will assume that any change in this consumer value actually reflects a change in trophic position when it may actually be reflecting a change at the base of the food web from primary producers. So just to give you a brief example, of some of the things that really impact this are events like upwelling. So deep ocean nitrate tends to have a relatively low stable isotope value of about 5 per mil, and it's pretty constant. So if you have big upwelling events, that has the potential to change the distribution of nitrogen in your water column, and thus what is being sequestered by those primary producers. Similarly, things like discharge can bring things like anthropogenically derived nitrogen into coastal food webs. One example here in the Puget Sound is things like wastewater treatment. That tends to have a really high enriched signal. So if a lot of wastewater is entering a certain area, that can impact what's getting into primary producers. And as a result, that's going to impact what isotopes are actually in consumer tissues as well. So variations in physiology of the consumers we're interested in also have impacts on our stable isotope value. More specifically, they have impacts on this thing that I referred to earlier, the trophic enrichment factor. So one example of this could be something like diet quality. If you're measuring stable isotopes in a fish consumer, and they're consuming another fish, that would be considered like a high quality diet, and you wouldn't expect there to be a very high trophic enrichment factor. So there wouldn't be much of that change between their diet and their own tissues. And that's just a matter of that the proteins they're consuming are very similar to the proteins that they actually need in their own tissues. In contrast, if you have a fish that's eating something like zooplankton, you can expect that that trophic enrichment factor might be a little bit higher. And similarly, things like somatic growth and disease and the specific tissue that you sample can also have implications for how big that trophic enrichment factor usually is. Regardless, though, what tends to happen in a lot of stable isotope studies is that a single universal trophic enrichment factor of 3.4 per mil is usually applied, and it's assumed that this is accurate for most consumers. However, we know from a number of different controlled feeding studies that this value of 3.4 per mil might actually not reflect common trophic enrichment factors. So this graph that just popped up shows 3 consumers differentiated by the different colored bars. So we have guppies, zebrafish, and minnows that were fed 3 different diets. So a diet of shrimp, perch, and swordfish. And you see here on this red bar that denotes our 3.4 classic trophic enrichment factor. And one thing that should really stand out is that for the most part, most of those species on those different diets don't have a trophic enrichment factor that is very close to 3.4 per mil. So for example, if you were to measure the isotopes of a guppy that's eating a perch diet, and you apply a 3.4 per mil trophic enrichment factor, when in reality it only has about a 1 per mil trophic enrichment factor, you could be overestimating that trophic or underestimating that trophic position by almost an entire trophic level, which is pretty substantial. So the final thing that I want to talk about before I get into the case study too much is one other variation in physiology that's really important. So the other thing is when a consumer is eating some kind of resource is those resources are not instantaneously incorporated into their tissues. So for example, your consumer might eat something on day 0. It might take about 5 days for that to be incorporated into their blood. It might take a little bit longer, like 20 days for it to actually be in the liver, and something even longer, like 80 days for it to be incorporated into the muscle. So if you're measuring your primary producers in your study and you're measuring your consumer, sometimes we could account for that in our sampling strategy. So for example, you could measure your primary producers on day 0. You know something about the tissue turnover time in blood is about 5 days, so you measure your consumer a bit later. You might be able to account for the tissue turnover time. And if you have a pretty resident non-dynamic system, that might work relatively well. However, if your animal moves on day 10 or day 45, it migrates somewhere else, or prey switches on day 10, that's a lot to account for in your sampling strategy. And I would argue if you have enough information to know when an animal is both prey switching and migrating to target your sampling strategy to account for all these things, you're probably not going to be deriving a lot of extra information from measuring stable isotopes to begin with. So to just kind of summarize this a little bit before we move on, it's really important that we measure the stable isotopes of the base of the food web, specifically in dynamic systems. And applying a single trophic enrichment may actually introduce error into our trophic position calculations if we don't think about it with the way that we're parameterizing our equations. And then finally, it's important that when we do measure primary producers and consumer tissues, that we make sure that those are spatially and temporally coupled, so that we're thinking about things like migration or so that we're accounting for things like migration. And now that I've hopefully convinced you that stable isotopes are really hard to apply and they actually have a lot of biogeochemical and physiology variations that we should be thinking about, I'm going to talk a little bit more about some approaches that we can take to kind of make them a bit more informative and account for some of this variability. So first, measuring stable isotopes in individual compounds, specifically amino acids, can be really informative and really useful for controlling some of this variation. And second, if we really carefully parameterize our trophic position equations, we may be able to eliminate some of the variability that's introduced by things like changes in trophic enrichment factor, for example. All right, so I'm going to start getting into some of the harbor seal data for this case study now. And specifically, I'm going to start with how we calculated trophic position for these harbor seals and some of the things that we needed to think about for applying and actually calculating these trophic position data. So first, I want to introduce you guys to this idea of amino acids and specifically what they do with applications of stable isotopes. So there are these groups of amino acids called source amino acids. One of the most commonly used one is phenylalanine. And source amino acids have this unique characteristic where they don't enrich up the food web like we see when we have more classically applied bulk stable isotope data. Instead, the value that is sequestered at the base of the food web by primary producers is conserved with each trophic transfer. And what that ultimately means is that the stable isotope composition of phenylalanine and primary producers is similar to that in something like a top consumer like a harbor seal. So what this means is if we measure the stable isotope composition of these source amino acids, it eliminates some of the challenges with trophic enrichment. It also directly measures the baseline from consumer tissue. So what that means is it's basically metabolically coupled with the resources that are ultimately getting into the food web and that are ultimately assimilated into those consumers that we're interested in. So we don't have to worry so much about these things like prey switching or migration because it's kind of like an internal control for some of those because of the metabolic coupling. Harbor seals are also really interesting consumers to be looking at and applying these types of data for. First, they're really generalist, so they're integrating over a number of different resource pathways, particularly here in Washington. They also consume a lot of economically and conservationally interesting species. So for example, harbor seals consume a lot of Pacific herring, and in about the 1970s, harbor seals in Washington increased their populations exponentially in response to the Marine Mammal Protection Act. And during that same time period, we've seen pretty dramatic declines in Pacific salmon, and a lot of that could be accounted for or could be because of this increased predation on these salmon species. Similarly, they also eat really high amounts of things like Pacific herring and hake, which are also economically important here. Some other important things to think about when applying these data for harbor seals is that they tend to have really limited migration and high site fidelity. So we know they're not utilizing resources in vastly different locations. They tend to forage 5 to 10 kilometers approximately from their haul-out sites and at depths less than 200 meters. So when using things like museum specimens, we can be fairly confident that the area that the harbor seals were recovered from was a similar area to where they were feeding on. Of course, that does have some variability, but we can be confident that a harbor seal collected here in the Salish Sea wasn't feeding in the Gulf of Alaska or off the coast of California or something like that. There are some evidence of juvenile males migrating a bit, but not on spatial scales that we're really concerned about. More importantly, from the chemistry standpoint, there's also been controlled feeding studies of captive harbor seals. So there have been studies that have specifically looked at the trophic enrichment between source amino acids and what I'm going to get to in the next slide of trophic amino acids. So we know these trophic enrichment numbers specifically for harbor seals, so we don't have to imply a broad number to them. So as a result, they really tend to be optimal consumers for this type of interpretation. All right, so what did we actually sample? We sampled 150 harbor seal skulls that have been curated from 6 different museum institutions across the US and Canada. And within those skulls, we measured the stable isotope composition of 1 source amino acid, phenylalanine, and 9 trophic amino acids. So trophic amino acids behave a bit more like I talked about earlier in some of the earlier slides, where they do show enrichment up the food web. So we can combine both trophic amino acids and these source amino acids to actually get our trophic position calculations. One of the challenges with museum specimens is that when it comes to sampling, you kind of have to take what you can get. So in an ideal world, we would have been able to sample a standard number of specimens from, from 1928 to 2014. That's our sampling range. And have specimens from, every year, every other year, or something like that. But unfortunately, our data are quite gappy because what we're able to sample is basically at the whim of what some scientist or someone out beach walking happened to see or collect, 50 years ago. So we were able to get multiple specimens for some years, but not all years. And we also have some pretty large data gaps, particularly early on in our time series. So the 1950s was a time period where we actually had pretty limited data. We sampled specimens from across the Salish Sea and in coastal Washington, and we divided them into those 2 locations. All right, so when it comes to calculating trophic position from these amino acid data, it's pretty similar to how we calculate trophic position from bulk stable isotope data. So you take the stable isotope value of a trophic amino acid, you subtract the stable isotope value of a source amino acid, and then you also account for fractionation between trophic and source amino acids that actually occurs in primary producer tissues. So that's what we call this beta value here. Because we're subtracting that fractionation, we also add 1 to account for the fact that those primary producers are in trophic level 1. And then we divide it again by some trophic enrichment factor, similar to our regular equations. One thing that has come about with the application of this type of amino acid data is a similar pattern to what has happened in bulk stable isotope studies, where there tends to be this classic value for trophic enrichment factors of 7.6 per mil that tends to be applied in most studies. Similarly, there's a classic beta value, which is reflective of the beta value of diatoms, which is about 2.9 per mil. Unfortunately, we know from that controlled feeding study of harbor seals that the trophic enrichment factor between harbor seals and their prey is much, much lower than this classic value and is actually closer to 4.3. Similarly, some other work that we have done with these harbor seal specimens has found that C4 plants actually contribute pretty substantially to Salish sea food webs, as opposed to it being exclusively similar to diatoms. And those C4 plants have a beta value of negative 8.7 as opposed to this positive 2.9 value. So when it comes to calculating trophic position, that's a pretty substantial difference if some of the resources that are being assimilated into this food web actually are reflective of C4 plants, even partially, as opposed to exclusively C3 plants. So what that means is we have some decisions to make about how we should parameterize trophic position. Specifically, we need to think about which beta values we should use, We could use an exclusively diatom C3 plant value. We could use an exclusive seagrass value, or we could try and derive our own value that accounts for the contributions of both C3 and C4 plants. Similarly, we need to think about how we should incorporate different trophic enrichment factors. So we have this harbor seal specific trophic enrichment factor. We have this classic value that tends to be derived from low trophic level consumers, or we can even use an average value that has been calculated across multiple taxa that is from a meta-analysis of amino acid papers. And finally, we need to think about which amino acids we should use. Now, typically, these studies use glutamic acid and phenylalanine only. However, this same meta-analysis by Jens Nielsen, who is a former UW postdoc, actually found that applying and incorporating more than one amino acid into your calculations overall improves your estimates. So another thing we need to consider is about whether or not we want to incorporate multiple amino acids. All right, so we're going to see a couple plots similar to this that are going to be parameterizing our trophic position calculations in a few different ways. So you'll see here on the top of each panel, we have an equation, and this is our trophic position equation. And the main thing that's different between each of these panels is how the trophic enrichment factor is applied. So in this first panel, we see just the trophic enrichment factor. In the next panel, panel 2, to the right of the first one, we see multiple trophic enrichment factors. We also see here these different colors are referred to the density distributions of trophic position of harbor seal calculated from that equation that's above the panel. And on our x-axis, we have trophic position. Finally, this gray box shows what is considered an ecologically realistic trophic position for harbor seals. So we'd expect them to be somewhere between 3.5 and 5. And this number here shows you the number of specimens that actually fall within that ecologically realistic range. All of these equations and all of these parameterizations also apply a diatom only or a C3 plant only beta value. And one thing that should stand out to you is that these trophic position calculations are pretty horrendous. Not very much of the data falls within that ecologically realistic range. So here you can see in this box, it shows that only about 15% of that data are falling between that 3.5 and that 5 trophic position calculation. Now for equation 2, we see a little bit of an improvement. We would expect that because we know there's at least one trophic transfer in this food web between harbor seals and their prey, but an improvement to 22% really isn't sufficient. In the next panel down, number 3, we see a bit more of an improvement, and this one performs best with our diatom only beta. And this incorporates that classic trophic position, trophic enrichment factor, and the kind of classic average value. And we get 66% of the data following within the range that we were expecting. But if we include both that low trophic level consumer trophic enrichment factor and the harbor seal trophic enrichment factor, we actually find that our calculations underperform rather than compared to the one that only has those low trophic level species. So overall, what we're finding with this particular parameterization is that we're not getting calculations that are sufficient or particularly useful. So it's a bit of back to the drawing board at this point. This particular plot comes from another data set that we derived from harbor seals that was part of another component of this project. And this is showing the stable isotope composition of carbon on our y-axis. And over here to the right, we see in blue, specifically Salish Sea harbor seal specimens, and in purple, we see coastal Washington specimens. Now, the important thing here to notice is that this green bar, anything above about -10 per mil, is close to what we'd expect for something like a C4 plant. Anything that falls about -18 to into the -20s, we'd expect to be a C3 plant. And as you can see, none of our harbor seals are falling exclusively in one of those ranges. And instead, what's happening is they're falling in between both C3 and C4 plants. So what we decided to do was to use this carbon data to actually calculate the contributions of these two different marine resources into the food web in which the harbor seals are foraging. And we can do this by applying a pretty classic stable isotope technique that's called a mixing model, where we take our carbon data for harbor seals, we subtract the C4 plant end member, which is basically the carbon value we'd expect for C4 plants, and then we divide it by the difference between the C3 plants and the C4 plants, so that we get this percent C4 that is contributing to the harbor seal tissues. You can rearrange the equation and get percent C3 too. I just happened to calculate it using percent C4. And then what we can do is actually calculate a beta value that is weighted for the contributions of both C4 and C3 plants into those harbor seal tissues. So if we go back and apply these equations again, using a weighted beta value instead of a diatom only value, we find that we get a pretty big improvement of the trophic positions that we're calculating. So if you remember from this first slide, this first panel was only having about 15%. We're falling within that gray box, or that ecologically realistic range. And now we have about 65%. And if moving over to the next panel, including that enrichment factor for harbor seals really improves our data, and we get about 76% falling within that range. By removing the trophic enrichment factor for harbor seals and including just those low trophic level consumer trophic enrichment factors, we perform a little less well. And then finally, the best model includes both those low trophic level consumers and harbor seals. Now that being said, there wasn't really a substantial difference between these two, and we decided to go with equation 2 instead of equation 4, just because it's a bit more parsimonious with one less parameter in that model. But overall, what we see here is that including this weighted beta really improves our trophic position calculations. One other thing that's helpful to notice is that not all amino acids perform exactly the same. And one thing that really stuck out to me when I was looking at these data is that specifically aspartic acid, this kind of light blue one, has this really broad distribution that is pretty different from a lot of the other amino acids. Now, one of the reasons that I think this is because aspartic acid tends to have a really low tissue turnover time, specifically relative to phenylalanine. So when an animal consumes something, it takes nearly twice as long for that aspartic acid to be incorporated into its tissues compared to the phenylalanine. So what I think we get, why we get this weird distribution is just because of those differences between tissue turnover times. So now kind of answering some of these questions that we had before, we have decided to use a weighted beta because it drastically improves our trophic position estimates. We have used a combination of 2 different trophic enrichment factors, so this average value and the value of harbor seals. And then we also decided to eliminate aspartic acid from our trophic amino acid data set and use only these 4, so glutamic acid, alanine, valine, and proline. And we're using only one source amino acid, which is phenylalanine. Now, one other thing to consider about about tissue turnover time, other than the differences between different amino acids, is to also think about how long it takes those stable isotopes to actually be incorporated into bone tissue. And there actually really aren't very many great studies about this. One reason being that to actually measure tissue turnover time usually requires multiple samples of tissue throughout time after you've prey switched for an animal in a controlled setting. And it's obviously very hard to measure bone in a living species. So there really only is one study with birds that has shown this. So we expect the tissue turnover time to be somewhere around a year based on those studies. So what that means is that the bone tissue of harbor seals, you know, if we have a skull that's from 1977, is actually reflecting the ecological condition that existed that previous year, because it's taking time for that actually to get assimilated into the bone tissue. So we also decided to apply a lag to our data before to account for this kind of physiological delay that exists because of this tissue turnover time. All right, so now hopefully I'll get into some of the more interesting questions, or at least more interesting applications for people who perhaps don't use stable isotopes. Thinking about how harbor seal trophic ecology responds to both environmental change and changes in prey availability. So to get at this question, we started by compiling a number of covariates that we thought would impact the food web in which harbor seals forage. We also were interested in including environmental covariates that impact things like the biogeochemistry in the system as well. So we included upwelling, an index of coastal upwelling, and we had an average for both spring and summer. We included sea surface temperature, discharge of both the Columbia River and the Fraser River, although we ended up not including the Fraser River just because the two variables covariate a lot. And then we also included climate regime. So specifically thinking about things like the Pacific Decadal Oscillation, the North Pacific Gyre Oscillation, and the El Nino indices. Now we are also interested in the food web component, so thinking about prey covariates as well. The main limitation when it came to compiling prey covariates was trying to get time series that really overlapped a lot with our harbor seal data, especially because our harbor seal data is pretty gappy. So we wanted covariates that really extended before 1970, which was a bit challenging. Regardless though, we were able to use things like Chinook escapements, coho escapements. We used an index of wild Chinook smolt production that was calculated with some of Brandon Chasko's work. And then we also used hatchery Chinook smolt production. We were interested in thinking about things like chum salmon as well, but unfortunately the data sets just didn't overlap as much as we were hoping to get like a robust analysis. We are also interested in herring and hake, mostly because these tend to make up a much higher percentage of harbor seal diet than Pacific salmon. We're a little unsure about whether or not we'd actually get a relationship between harbor seal trophic position and salmon, just because they tend to be a relatively small component of the diet based on scat samples. And the herring and hake data were both spawning biomass. Now, one of the other thing that we were interested in was also that there tends to be a lag between environmental change or ecosystem change and the response of predators. So for example, there's been evidence that things like northern anchovy abundance tends to have a lagged response to changes in ocean temperature. Similarly, breeding seabirds tend to have a lagged response to changes in oceanic conditions. So we wanted to consider this as well in our analysis. So in addition to including a lag for this physiological delay, we also included a lag for an ecological delay as well. So this was a lag of 2 years, which incorporated both the physiological delay and a 1-year delay in the ecological response. And then we also included a lag of 3 years. So this was a 2-year delay in the ecological response and a 1-year physiological delay. So initially when we started compiling this data set and trying to sample museum specimens, we really kind of were approaching it thinking that we were going to have some kind of classic time series analysis, you know, perhaps a Mars model or a dynamic factor analysis. But some of the data challenges, specifically the large temporal gaps and the kind of tricky component when it comes to the time series of having more than one observation at one point in time, kind of ended up changing our plan, and I ended up applying just a hierarchical modeling framework where we used both the environmental and prey availability covariates in addition to the lags. So we had our environmental model, which included 3 separate models. The 1-year lag model that was just the physiological delay, and then the 2- and 3-year lag models that also included the ecological delay. We included all possible combinations of environmental covariates, including up to 3 covariates in the model. And then we also had a random effect, which accounted for how trophic position was calculated with the different amino acids. So it was essentially a random intercept based on the different amino acids that was used to calculate it. Then we also had a food web model, which was basically the same modeling approach, where we had a hierarchical model using an amino acid as a random effect, and then we had different prey availability for covariates. All right, so now some of these results. So here in this plot, we're showing the best environmental model. These models were compared using AICC, and then all of the models that were considered our best model, we also just checked to make sure we didn't have high multicollinearity using things like variance inflation factors as well. But for the most part, most models only included 1 or 2 covariates as well. Things like covariation between our covariates didn't end up being a major concern because we didn't have multiple covariates in most of the supported models. You can see here all of these models, regardless of the lag or delay that was applied, incorporated a location. I forgot to mention on the previous slide that we also had location as a factor. So the location factors were either Salish Sea or coastal Washington. And what we found is that harbor seals in the Salish Sea actually eat about 0.3 trophic levels lower than harbor seals in coastal Washington. For our physiological delay only model, we found that summer upwelling was an important determined harbor seal trophic position, and that there was a negative correlation between the two. Oh, sorry, I forgot to tell you what all the axes were. So here on the right, we have our covariates. Here on the left, we have the actual coefficient estimates. In the black on this plot are our fixed effects, so those are essentially just our covariate values. And then here in these colored values are our random effects for the different amino acids. So for this physiological delay only model, this is essentially showing you like upwelling in 1977 impacted harbor seal trophic position in that same year. So for our ecological delay model, we're seeing a delayed ecological response. So summer sea surface temperature in 1976 impacted harbor seal trophic position in 1977 is essentially what that's showing. And we actually have that these two are negatively correlated, indicating that with higher sea surface temperature, harbor seals are actually feeding lower in the food web. And finally, for our 2-year ecological delay, we actually see that amino indices have a negative impact on harbor seal trophic position, along with summer upwelling, which has a positive impact. One thing that's worth noting is that summer upwelling for both the physiological delay only model and the 2-year ecological delay, actually, while it differs significantly from 0, it's not a very large trophic level response. So as you can see here, the covariate's around 0.04. So while it is statistically significant, it really doesn't have a large ecological significance. It's the only difference is a trophic level about 0.04, which is very substantial. All right, for our food web models, I found these ones to be a little bit more interesting. For our physiological delay only, we had a significant impact of hake spawning biomass abundance on harbor seal trophic position. So this is showing that in years with high hake spawning biomass, harbor seals are eating higher in the food web. And this is pretty intuitive because hake tend to be a relatively high trophic level species for harbor seal to be feeding on, specifically adult hake, which we would expect the spawning biomass covariate to be reflecting. For the 2-year or the 1-year ecological delay model, we found that Chinook smolts actually had a positive relationship with harbor seal trophic position. And this was really confounding to me at first, but hopefully I will explain it in the next slide to you all why this is a positive correlation. And this Chinook smolts covariate actually was a combined index of both wild production and hatchery smolt production. And then finally, in our 2-year ecological delay, we see some other changes. Again, we have this positive correlation with Chinook smolts. We have a negative correlation with hake spawning biomass. And then we also have a positive correlation with herring. And again, we have very similar support for this location factor as well. And the overall approximation of that factor is pretty similar between our food web models and our environmental models. All right, so why are Chinook smolts resulting in a positive trophic position correlation with harbor seals? For me, when I first got this result, it was a little bit counterintuitive. Chinook smolts are pretty small. If harbor seals are eating more smolts, we'd expect them to be eating lower in the trophic level. And I think it's because of the more delayed framework that we have this data modeled in. So in these boxes are the covariates that had significant support in our models. And what I think is happening here is that Chinook smolts are contributing in the previous 2 years and in the previous 1 year are contributing to the current abundance of 1 ocean and 2 ocean salmon that are available to harbor seals. Now, it was really interesting to me also that these small indices were more significant than using escapement numbers, but it also makes a lot of sense, right? So escapement numbers are the number of Chinooks that are re-entering freshwater after they've already been predated on and after they've already been fished. So it turns out that these estimates of Chinook smolt numbers seem to be better predictors of what is available to harbor seals, and as a result seem to have a higher correlation or higher importance in determining harbor seal trophic position than using an escapement indice alone. So stepping through some of the other data we found this positive relationship between hake spawning biomass and harbor seal trophic position. This is expected. Adult fish should be producing a higher trophic level, especially hake, because they tend to be bigger and one of the higher species that harbor seals are feeding on trophically. However, we get this negative response when it comes to including hake spawning biomass from 2 years ago. And I suspect that this is because hake spawning biomass from 2 years ago is determining the current juvenile hake abundance numbers. And harbor seals are also opportunistically taking advantage of that prey source as well. Because one thing to think about with trophic position is that it's not just a matter of which species an animal is feeding on, but also some of the life history or the life stage of those individual species that are going to impact what trophic position they are in. And then finally, I think we are seeing a similar thing with herring, where the herring spawning biomass from 2 years ago has a bigger effect on current harbor seal trophic ecology than the current spawning biomass. I'm still kind of parsing out the results of this particular part and why we're getting this positive correlation. So if anyone has any thoughts on this, I would certainly welcome feedback. So that's about all I had, just kind of in summary. Careful decision of parameterization can really lead to a more informative analysis, especially when you start to consider things like tissue turnover and delayed ecological responses. And finally, prey covariates that don't represent availability to predators may miss some of the important relationships that we're seeing in this data. And with that, I would just like to thank my collaborators. We worked with a lot of different museums from kind of all over the place for this project. The first one we really worked with was the Burke Museum, and they were just incredibly helpful with helping us get the chemistry up and running for this type of data analysis. But we also worked with the Slater Museum at the University of Puget Sound, the Royal BC Museum in Victoria, the University of Alaska Museum of the North, Marine Mammal Laboratory at NOAA, and also the Smithsonian Institute in DC. And I also should acknowledge that most of this project has been funded by Sea Grant, and they've been great to work with for this work.
All right, thank you, Megan. Thank you for the great talk. We have time for questions. So if anyone in the audience has a question for Megan, you can go ahead and enter it into the chat, and we can read it back. Or if you prefer, alternatively, you can use the raise your hand feature, and we can unmute you, and then you can ask Megan your question. Let's see, I think. There we go. Let's see here, first question. Okay, Gordon asks, if compound-specific stable isotopes data are so great, why doesn't everyone use them?
Yeah, that's a great question, Gordon. So one of the reasons why anyone who knew me in my first year of grad school probably already knows the answer to this question. And the one of the big reasons is that they're incredibly hard to do chemically. So for example, for me to get 6 samples run, it takes like probably 4 pretty full days of lab work. Whereas, you know, in one day, you could probably pack well over 100 bulk stable isotope samples. There's also not a lot of labs that do this. There's only a handful of labs that really have the chemistry to do the compounds specific stable isotope analysis up and running. And it's becoming more prevalent. It is somewhat of a new tool. So I think within the next, you know, decade or so, it'll hopefully be cheaper and easier to run these types of samples, but it's really not at that point yet. Yeah, so the chemistry is definitely very, very challenging in running these samples compared to bulk samples.
Okay. Well, I think your talk was so comprehensive that you've answered everyone's questions already, Megan.
I have a question, Andy.
Okay, Sean?
Yeah, I can't figure out how to raise my hand as a organizer. Okay. So, Megan, have the how does the trophic position of the prey of harbor seals change over time in response to those changing environmental conditions?
Yeah, hold on. I have a slide that I can show you. So generally speaking, Sean, we don't really get a lot of huge changes through time. What we get more is like year-to-year variability. So once you account for the changes in the environment or the changes in prey, you tend to just have like a flat line in the trophic position. So there isn't really a general trend over the past century of harbor seal trophic position. I don't know if that fully answered your question. I thought a slide of the residuals after I added in the covariates, but I don't.
Thanks.
Okay, Megan, well, it looks like that is it for questions. So great
All right,
talk.
Andy, can I ask a question?
Oh, there we go. Oh, Helena.
Hi, Andy and Megan. This is Helena. I'm curious about why upwelling. Maybe you already explained this, Megan, but I'm curious why upwelling in the El Nino index is correlating with these seal trophic levels.
Yeah, so I think there's actually been a lot of evidence that thing, particularly El Nino, really impacts prey availability. So the mechanism for the environmental impacts on trophic position is basically the environment is impacting some availability of prey, and then that's impacting trophic position. We were interested in looking at environmental covariates because the idea being that they probably have impacts on like broad amounts of prey as opposed to one individual species. So when we were first doing this analysis, we weren't really sure if we were going to see responses to individual species or if it would be like more determined by environment that's impacting a lot of species at once. So it has been shown that El Nino does impact prey availability. Generally, it has an impact in the California current. But yeah, so my guess is what's happening is that the ocean conditions are impacting what's available, and that's impacting trophic position.
Okay,
I'm not
thanks,
sure
Megan.
that fully answered what you were asking, but
Yeah, no, that does answer it. And then to follow up, are you sort of thinking that warmer years or lower upwelling is leading to lower food, like lower production, lower food availability? So they're maybe having to, go for a less nutritious, maybe lower trophic level food source?
yeah, and it's hard to say exactly, because when you think about trophic position, it's like a little bit of mental gymnastics. But yeah, so both of those tends to be negatively correlated. So the idea is that they seem to be taking more advantage of lower trophic level species. So that might be juvenile species, or it might be something more like eating a higher proportion of something like forage fish in their diet as opposed to adult species.
Okay, cool. Thank you so much. I really enjoyed your talk. I learned a lot, and it was amazing to see the difference in what was expected and what you found in your recent paper that I'll look up later, of the proportion of the primary food source that is coming from seagrass, and that value is so much lower than what was expected. So that was really interesting.
Yeah, thanks.
Thanks, Helena. Okay, I guess looks like that's it for questions, Megan. Thank you for the great talk.
Yeah, thanks for inviting me.
Well, everyone, thank you for joining us. And thank you, Megan. And I hope you all join us again next week, where our speaker will be Sarah Converse, giving a talk, Synchrony in Seabird Survival, Drivers at Multiple Spatial Scales. So thanks again, everyone. And thanks again, Megan. And we hope you all have a wonderful weekend. And we look forward to seeing you next week.